Hey guys, this is me Akash here for Biology Vibe. So today we're gonna start Vibe 1.6, where we're gonna discuss about the central dogma of biology and a little bit about genetic code. So, as we all know, DNA is the boss. It's the actual genetic material which is governing the whole cell and our body in whole. So, what happens is like now just think about your body. Your body is mainly made up of proteins, your enzymes, antibodies in your body, your hair, nails, skin, everything is protein, right? So, what things determine the structure of that protein, the function of that protein? Definitely, its constituents, that is amino acid, and the type in which they are arranged, the sequence of amino acids. And what will decide the sequence of that amino acid? Our DNA. So this is the story is all about. We all are made up of proteins. The proteins are made up of amino acids, and the structure and function of all the proteins is determined by the sequence of that amino acid, and the sequence of that amino acid is determined by our DNA. So we say that yes, DNA is the actual boss, and DNA is basically here, that is in the cell's nucleus, and it has to send signals to the protein factories of the cell, that is the ribosomes, to put the exact match of that amino acid which the DNA is coding for. So the whole process is known as the central dogma. Now, the DNA will definitely not move from inside the nucleus to the protein factories and tell them that hey, please put this amino acid first, this second, this third, this fourth, and stuff like that. So definitely it will pass this code, make some molecule, and then that molecule will go to the protein factories of the cell, that is ribosomes. So this red ribbon which you are seeing, this is called the mRNA. This mRNA is being made from the DNA via some process which is known as transcription. So basically the codes which are written in this DNA makes a copy to this mRNA. This mRNA is known as the messenger RNA. It moves through the nuclear pore and goes to the ribosomes. The ribosomes accepts it, may just get the exact sequence from where it has to start the further process to put on all the amino acids in a sequence. Now this process is called translation. So I just use two main terms, transcription and translation. Transcription refers to when DNA is coding for the mRNA, and when the mRNA is being decoded in the ribosomes, it's called translation. The central dogma is all about transcription and translation. So this was a kind of overview what central dogma is. We'll discuss transcription separately and translation separately in different videos in details. So now I'm just saying that DNA has some codes. Now that codes are in the form of genetic code. Now what is that? Let's have a basic understanding of what genetic code is. We all know that DNA is made up of nitrogenous bases, that ATGC sequence. So a set of adjacent three nucleotides makes up a codon, and each codon is signaling a specific amino acid. Now let's get into details of this. First, let's start with the definition. The set of DNA and RNA sequences that determine the amino acid sequences used in the synthesis of organisms' proteins. It is the biochemical basis of hereditary and nearly universal to all organisms. So just have a look at this diagram. This is the DNA. The DNA codes for mRNA. So now see, this is the mRNA. It will move to the ribosomes, right? So it has some codes written. ATGCTAGGC. For example, this is the code. So a window of 3, that is ATG, will signal a specific amino acid, this yellow one. CTA will signal a specific amino acid, pink one. GGC will signal a specific amino acid, this blue one. So the sequence, that is, first the yellow one amino acid will come, then pink amino acid will come, then blue amino acid will come. This is as per our genetic code. So you see how smartly things are going. The specific genetic makeup is defining the specific sequence of amino acid, and the specific sequence of amino acid is determining the function and structure of a protein. In short, that DNA is indirectly making us. Now the question is, why is this a window of 3? Now there are 4 bases, A, T, G and C, and the amino acid number is 20. There are around 20 amino acids. So just one amino acid, if just one nitrogenous base will signal one amino acid, this way we'll able to signal only 4 amino acid. For example, if it's double, that is doublet. For example, AT, GC, TA, GG, like this. Then also it will make a combination of permutations of 16. So 16 codons and 20 amino acid to be signaled. Then again it's a problem. Now keep it 4 to the power 3. Now 64 codons are there, like this 64 unique windows are there, and they have to signal 20 amino acid. So this is good enough. If you keep it 4 or 5, the number of permutations will be much larger, 256, for signaling 20 amino acids. That's huge, right? So it's not 4, it's not 5, it's exactly triple. So the first property is that the codon is triplet. Now the second thing, it's non-overlapping. For example, if you see AUU, GCU, CAG, this is the sequence. So now the code is non-overlapping. That means it's 3 discrete units. AUU, that's all, finished, one codon done. Then GCU, second codon done. It's not like that the upper one, it's not overlapping, it's non-overlapping. It's not like AUU and the second codon becomes UUG and the third codon becomes UGC. It's not like this, not overlapping, it's non-overlap. It's not overlapping, it's non-overlapping. Okay, so this discrete units, AUU signaling one amino acid, GCU signaling second amino acid, it's discrete, different, and specific windows are there. So it's non-overlapping. The code is commaless, it's written in simple straightforward form, AUU, GCU, CAG, CUU, like this. No commas, no punctuations, like that. Now it's non-ambiguous and degenerate. Now, non-ambiguous means, for example, if a codon in the mRNA becomes CTT, it will signal leucine. It will not signal anything else, it will signal leucine. It's clear in its signal. If it says AAT, that's a specific signal. If it says AA, that's a specific signal. For example, if it's at CGT, it's arginine, that the specific amino acid is signaled. So it's non-ambiguous. Different codons have specific, they just signify specific amino acid. Non-ambiguous. Now the second thing is degenerate. If you are able to see, there are 64 codons and they have to signal 20 amino acid. Now let's consider an example here. For example, you are driving a car. So the signal is red. So you know you have to stop, right? If the police is holding that rod, if the traffic police is holding that rod and it signals you red light, then also you know you have to stop. If nothing is there and the traffic policeman is just waving that hand and showing that you have to stop, then also you will stop. So if you have the signal over you, if the signal is in the hand of the traffic police, and if just via hand the traffic police is signaling you to stop, you will stop. All these 3 signals will say you to stop. Similarly, if it's CTT, CTC, CTA, CTG, anything, or TTG or TTA, any of these six codons, it will signal leucine, right? If it's TAT or TAC, both will signal only tyrosine, right? So more than one codons are there that are signaling specific amino acids. This is the point. This is known as degenerate. Now, if you are able to look here, there is TGA, TAA, TAG. These three codons are known as stop codons. Why so? These codons, when comes in mRNA, they signal the protein synthesis to be stopped. That means they have no corresponding amino acids. So when TAA comes into the mRNA, the ribosomes know that there is no amino acid which is coming for them. So definitely the protein synthesis have to be stopped. So these three are the stop signals: TGA, TAG, and TAA. So that's kind of it, guys. In Vibe 1.7 we are going to discuss transcription in details. So if you like the video, please hit the like button, share this video with your friends, and definitely subscribe to my channel to get more and more videos like that. Thank you.